we are connected with Professor John Harris. John Harris is Chair of Dermatology and Director of Autoimmune Therapeutics Institute and Director of the Vitiligo Clinic Research Center in the University of Massachusetts Medical School. He is well known to everyone working on vitiligo, of course. He is a physician, a scientist caring for patients in a vitiligo specialty clinic and running a research laboratory focused on understanding the disease pathogenesis and developing new treatments. He's also the founder or co-founder of several startups and companies that are dedicated to the development for drugs, treatment for inflammatory skin diseases. So it's really a pleasure that John is connected with us, and he will give the talk live. So basically, as we should be here with us. So please, John.
Hi, everyone. Thank you, Dr. Picardo. Appreciate the introduction, and I'm sorry to miss being there with everybody in Bangalore. It sounds like you're having lots of fun. And I have the, what we call here FOMO, the fear of missing out. I can see everybody in the audience, and I've been joining in when I can remotely. I know you've already heard excellent lectures about vitiligo pathogenesis from Dr. Picardo, Dr. Pandya, Harsad, Esmat, Passerone, and others, and you've heard about interferon gamma, JAK inhibitors, clinical trials. So I don't know that you need me so much, but I'm happy to talk a little bit about now what we've been working on in terms of vitiligo pathogenesis, really with an eye toward finding new treatments. These are my disclosures. There are a number of them on here. I work with a lot of companies to help them develop drugs for vitiligo. The main one to focus on here is Incyte Corporation. One of my companies developed a drug called auremolimab. They acquired that, and we're working with them now to bring that into the clinic, and I'll talk a little bit about that. So our research strategy in the lab to really study vitiligo pathogenesis with the goal of developing new treatments is to seamlessly integrate basic science in a mouse model, translational research in tissues from our patients, as well as clinical trials to develop an understanding of disease relevance, disease pathogenesis with relevance to clinical treatment. So here's a story of bench to bedside. I think you've heard this one before. We looked initially at gene expression in the skin of our mouse model in patients with vitiligo. You can see that there was a loss of melanocyte transcripts, as you'd expect to see in vitiligo. And then we saw interferon gamma expression, and then a number of genes that are induced by interferon gamma. So we saw this interferon gamma signature. We didn't see anything from type 17 inflammation or type 2 inflammation, and this is important because we have good biologic therapies for psoriasis and atopic dermatitis that target these pathways. This is why it appears they don't work for vitiligo, because they're targeting pathways that aren't turned on. In fact, we've seen that all of these drugs have induced or worsened vitiligo as well. So we needed to find an equivalent therapeutic to target this pathway. This is Mehdi Rashigi and Jillian Richmond, who are fellows in the lab. This is about 15 years worth of work in one slide. But we targeted the interferon gamma signaling pathway in our mouse model. We were able to knock out interferon gamma, the receptor, chemokines and their receptor, as well as STAT1 to show that this prevented the onset of vitiligo. We're also able to use antibodies against members of the pathway. This prevented vitiligo as well as reversed it and showed that this could be a treatment for disease. And we also found that JAK inhibitors were effective treatments in our mouse model. But maybe more importantly, they're effective in humans with vitiligo as well. And this is a study that we did with Angela Cristiano and her group in New York. Actually, I presented earlier today to the Alopecia Areata Summit. And so it's been a pleasure. I've been all over the world, here in my home and then in New York City today and now in Bangalore. But this was a patient who had both vitiligo and alopecia areata, and we found that oral ruxolitinib reversed his vitiligo and hair loss. Here you can see in about 5 months, significant repigmentation of his face, as you can see. And we had actually been following this patient for over a year. We've been collecting serum from him, and we found that his CXCL10 level was elevated in stable in his serum for over a year, until he started taking ruxolitinib, and then we saw this level drop precipitously. And this really told us that JAK inhibitors not only were effective in patients, but they seem to be working the way we'd hypothesized by blocking interferon gamma and downstream effects. So this and a study by Brett King at Yale using oral tofacitinib in a patient with vitiligo, as well as David Rosmarin in 12 patients with topical ruxolitinib, showing that all of those worked. This really provided the rationale to start clinical trials testing JAK inhibitors as treatments for vitiligo. This is Zeni Abbas, who was a fellow at the time, helped us run these trials. And you've already heard the results from the Insight and Pfizer clinical trials, just really phenomenal stories leading to FDA approval of topical ruxolitinib in our patients just this past summer. And after that announcement came out, I had about 300 phone calls from my patients asking for this drug. So we had lots of prior authorizations to do, but it's been great. We've been getting it, it's been available for our patients. We've been seeing great results. So we're excited about that. I won't give you that data because I know you've already heard it. So I'll tell you the next story of bedside prevention back. And this starts with observations in the clinic that vitiligo relapses when treatment is stopped. And most of you have probably seen this paper by Thierry Passeron, published in 2015, showing that vitiligo relapses at a rate of about 40% after stopping therapy. This can be reduced to under 10% with just twice weekly tacrolimus, and we use this all the time in the clinic. But just as interestingly, vitiligo, when it comes back, actually comes back in the same place it was before. So this is a patient that I treated with narrowband UVB. She repigmented her abdomen. She got pregnant, stopped the narrowband UVB, and within a couple of years actually relapsed just in this exact same location. And so that tells us that the skin where vitiligo occurs has memory. Somehow the skin remembers it's supposed to have vitiligo. And we wanted to know what the source of that memory was. So we went back to the bench to try to understand that. And we weren't the only ones. So there were 3 other labs from around the world that asked the exact same question we did. And you can see Julian Seneschal here. I know that you've heard his name many times during this meeting. And we all found that resident memory T cells are responsible for the maintenance and relapse of disease. Really showing that this is a pretty robust finding since we all came on the same discovery. CD8 T cells enter the skin, they find melanocytes, they make interferon gamma. That induces gamma-induced chemokines like CXCL10, you've heard this a lot as well, and that recruits more cells to the skin through CXCR3, and this drives the progression of vitiligo. Turns out that a subset of these cells transform into what's called resident memory. This means they upregulate adhesion molecules. They glue themselves into the epidermis and stay there, so that when melanocytes crawl out of the hair follicle to repigment the skin, they engage with these cells. They make interferon gamma again, CXCL10, recruit more cells to attack the melanocytes and maintain the disease. Now, this function can be blocked with conventional therapies and JAK inhibitors. Turns the resident memory T cells off so that the skin repigments. But when the treatments are stopped, these cells are still there, they wake back up and they reinitiate disease. And we saw this in our mouse model. You can see that the JAK inhibitors work to repigment the mice, but the resident memory T cells actually don't go anywhere. They're still there over time. And so we asked whether we could remove the resident memory. Could we erase the memory and provide long-lasting treatment effects that remain after the treatments are stopped? And Jillian Richmond, as a fellow in the lab, discovered that an antibody against IL-15 receptor beta could repigment the mice. This is actually a response that we hadn't seen in any other treatments. This was the greatest response we've seen. And importantly, the resident memory T cells, when we did this, also disappeared from the skin, unlike what we saw with JAK inhibitors. And we only had to treat for a couple weeks to see months of repigmentation. And so this told us that not only was this a good result, but a long-lasting one. So we're excited about this. And there are 2 ways to block IL-15. You can block the cytokine with an antibody. You can block the receptor as well. And Amgen actually has an antibody against the cytokine. And so we requested funding from the NIH through the Immune Tolerance Network to test their antibody as a treatment for vitiligo. This is an ongoing study called the REVEAL study. And I had written the protocol to do this study, and then had to recuse myself because I was able to receive venture capital funding to start Valaris Therapeutics in order to create an antibody against the receptor. There are a number of reasons why we think this is a much better approach and more likely to be effective in the clinic. I don't have time to talk about that today, but happy to discuss another time. So in about 3.5 years, we developed an antibody called the auremolimab. It's a humanized antibody that targets the receptor, and we had plans to be in the clinic next year. But we were, about a month ago, we were acquired by Incyte. We're really excited by this. We're happy to partner with Incyte to bring this therapy into clinical trials shortly, and then hopefully put it in the hands of patients soon. All right, so those are two pathways, interferon gamma signaling, IL-15 signaling, and we wanted to find out if there are other potential signals that we could develop treatments to target to develop better therapies. So we asked whether we could dig deeper into the disease with higher resolution to see what else is going on. So to help us do that, we optimized a skin blistering technique in patients using a negative pressure machine and blisters. Here you can see confetti vitiligo here on the foot where we blistered right over that, as well as control skin at a distance from that. So we could then access the skin cells from the blister fluid and do a number of assays to explore what's happening there. And here you can see we can take the cells out of the blister. We can do flow cytometry, single cell RNA sequencing. We take the fluid and we can look at ELISA, proteomics, metabolomics. So I'll tell you a little bit about our study with single cell RNA sequencing and what we were able to find. So this is, these are all the cells that we found in the skin. Each dot is a cell that we pulled out from about 10 vitiligo patients and 7 or 8 healthy controls. You can see we found keratinocytes, lymphocytes, dendritic cells. We could see macrophages, melanocytes from the lesions that were under attack at the time. So we're able to really learn a lot about this. And the keratinocytes, interestingly, we could see basal keratinocytes, granular keratinocytes, spinous, and everything in between. When we looked at the lymphocytes in more detail, we could see CD8 T cells, CD4 T cells, T regulatory cells, conventional cells, gamma delta cells, NK cells. And we next asked which cells were making which ligands and ligand receptors in the disease, and which of these connections were changing in lesional skin compared to non-lesional compared to healthy control. So this is Manuel Garber, who is my collaborator here in computational biology, and these are our graduate students, Jake and Jim. And you can see that we asked, of all the ligands being the human ligands in the human genome, we could see that there were a number being expressed, and these were very cell-specific. So you could see some ligands being expressed by some cells and different ligands by other cells. And then the receptors was the same story. This was very cell-specific who was making the receptors for these ligands. And you can imagine if we can connect a ligand with its receptor on each cell, we can visualize a number, infer a number of cell-cell communications. And these appear to be one-way communications from one cell making a ligand to another cell making its receptor. And we can see some conversations are going up over time in lesions and others are going down. And so we can take these communications and assemble them into a network where each dot is a cell type here, communicating with a different cell type. The edge in between them represents that communication, and the red edges are going up in lesions, and the blue edges are going down. So you can see notch signaling, TGF-beta signaling decreasing in lesions, chemotaxis increasing, class 1 MHC expression increasing. And we can look even closer within these groups within the network. We looked at chemotaxis, and here you can see the ligands on each cell type being expressed here, connected to their receptors on the cell types as well. So there's a lot of data in here, but you can see CXCL9, CXCL10, and the receptor CXCR3 being expressed here on macrophages, keratinocytes. So this really gives us confidence in the data, validates it with known pathways. And then we looked and found CCL5 expression, CCL5 being expressed mostly by CD8 T cells, and CCR5, the receptor for that, expressed on regulatory T cells and CD8 T cells. And this was increasing in lesions compared to healthy. So we looked a little bit closer at this, at this CD8 T cell Treg conversation. This is the expression of the CCR5 on all the cell types, just to show in another way. We can see it increasing in regulatory T cells and lesions. And we validated this using flow cytometry for CCR5 on Tregs, gating on Tregs from blister fluid in patients, and you can see that lesional Tregs expressed higher amounts, levels of CCR5 by protein as well. So this is Kingsley Essien, who's a grad student in the lab at the time, and he found the same thing happened in mouse vitiligo as well. So when we looked in vitiligo lesions or the lymph nodes of vitiligo mice, we could see increased CCR5 expression on regulatory T cells. So he wanted to look by imaging. He's got great hands in the lab and was able to make this work, where we looked at our mouse model where the Tregs expressed green fluorescent protein and the CD8 T cells expressed red fluorescent protein. And we could see these clusters of cells in the ears of the mice as they developed vitiligo. You can see the green and the red cells really clustering together. And he was also able to use confocal microscopy to look even closer at these interactions. And you can see the green Tregs and the red CD8s really almost shaking hands in close contact, in cell contact, really suggesting that this regulatory effect was through cell contact mediated mechanisms. We could see this in human skin as well. Again, the green Tregs with the red CD8s, we could see this close cell-cell contact, and we could see that the Tregs were expressing the CCR5 within human lesions as well. Really kind of recapitulating what we saw in the mouse. So next, he developed a mouse model of vitiligo where we could put in CD8 T cells and regulatory T cells, or to leave them out in a RAG knockout background. And we found when we put wild-type Tregs in, of course, we had very good control of the disease. So reduced disease. Here's Treg-controlled vitiligo on the ears with nice pigmentation compared to no Tregs where the ears turn white. And what we found was when we used CCR5 knockout Tregs, they were unable to control vitiligo to the same extent. And so you can see this here, increased disease compared to the wild-type Tregs. And our initial assumption was that CCR5 was probably responsible for bringing the Tregs into the skin. But when we did competitive comparisons where we put wild-type and knockout Tregs in the same mouse together and compared how well they were able to reach the lymph node and the ear, we found that the CCR5 knockout Tregs actually got into the ear skin just fine. This was a little bit surprising to us because we assumed there'd be a migration defect. So they were arriving in the ear skin, but they still weren't controlling disease. And so when we looked closer, Kingsley again this time took green wild-type Tregs with red CCR5 knockout Tregs, put them in together and imaged them. And you can see these clusters are forming, but there's a strong overrepresentation of the green cells, or the wild-type Tregs here, with very few red Tregs. And so the CCR5 knockout T cells, Tregs, even though they were able to get into the skin, they were not really going where they were supposed to, localizing with the CD8s. And this is a close-up of that with lots and lots of wild-type green Tregs, but very few knockout red Tregs. So this told us that the Tregs were not using CCR5 to get into the skin, but once they were there, they were using it to be able to localize with their targets, CD8 T cells. So we wanted to know what it was, what was the signal bringing Tregs into the skin in the first place. We can see that Tregs increase over time. This is from our single-cell RNA sequencing paper, that in lesional skin, Tregs are in higher number compared to non-lesional skin and healthy skin, so they're migrating into the skin. And we can see this in the mouse as well. So this is human. In the mouse, we see the Tregs going into the skin to try to control disease. So what was that signal? We initially thought maybe it was CXCR3. This is our older paper that Mady published in 2014 showing that CXCR3 gets effector T cells into the skin. It's responsible for their migration into the skin. And we find that the Tregs follow the effectors into the skin. So over time in the mouse model, the T effectors go in, CD8 T cells go into the skin, the Tregs closely mirror that. And so we assumed that they were probably following the same signal. But we were a little surprised and maybe a little frustrated to find that the CXCR3 knockout Tregs actually got into the skin just fine. They had no defect at all, and they were able to suppress disease just as well as wild types, indicating that they don't follow that signal. We had read this paper from a friend of mine, Mike Rosenblum, who studies Tregs in skin, and his postdoc at the time, Tiffany Scharschmidt. And they described the CCL20 chemokine signaling through CCR6 that promotes Treg migration during neonatal development. So we looked at this, and we found that the CCR6 knockout Tregs were completely unable to control disease. And when we looked this time, we could see that the wild-type Tregs outcompeted the CCR6 knockouts in skin migration, really telling us that CCR6 was the signal that the Tregs were following to get into the skin in the first place. So I told you a few stories here about JAK inhibitors, blocking interferon gamma signaling to treat disease. We talked about IL-15 signaling, the maintenance of resident memory T cells, and that as a viable target. And now Kingsley's work showing Treg migration into the skin as well as localization of CD8 T cells. And that CCR6 is responsible for this. Many have probably seen this paper that came out earlier this year in Nature describing the fact that also using single-cell RNA sequencing, showing that fibroblasts express CXCL9 and 10 at very high levels in the skin. Here you can see with more T cells, you see more chemokine. And the main take-home of the paper was that fibroblasts in different locations in the skin actually are more differently responsive to interferon gamma. So in these areas where they saw vitiligo more commonly, the fibroblasts were much more likely to respond to interferon gamma by making these chemokines versus other areas of the body. And so they were able to show that the more commonly affected areas of the body with vitiligo might be due to fibroblast induction of these chemokines and recruitment of the T cells. Really a fascinating study. So adding the role of fibroblasts to the CXCL10 story and recruitment of T cells, based on our single cell data and the excitement around this, we were able to get funding from the NIH to create a Vitiligo Center of Research translation. This just started in September, and this is a 5-year award to help us study even further, tying these pathways and cell-cell communications together, looking at T cell clones and how they drive disease in a collaborative way with our computational biologist colleague, Dr. Richmond, who's now on her own and independent at UMass, Dr. Kent, who studies autoreactive CD8 T cells, and Dr. Stern, who studies T cell MHC interactions. And then Dr. Long Kai at Caltech, who does spatial transcriptomics. So we're pretty excited to dig even deeper and deeper. Finally, I just want to leave you with a couple of examples of how we're using this information to develop novel therapeutics. I call this at the right place at the right time, both because we're working to deliver the therapeutics, the treatments at the right place at the right time, but also because I was able to collaborate with some key people because I was in the right place at the right time. So in 2006, Dr. Craig Mello, who is an investigator here at UMass, won the Nobel Prize for discovering RNA interference. And he's a close collaborator and good friend here at UMass. Anastasia Kvarova came from industry shortly after and started to work on RNAi from a chemistry perspective and to modify RNAi chemically in order to make it longer lasting in vivo and able to target certain tissues. And her work actually really won this Nature Biotech's 25 landmark papers in the last 25 years because of her chemical modifications of RNAi. So after Craig discovered RNAi, we thought that this was going to be a great treatment for lots of diseases. But the problem was RNA has a very short half-life in vivo and is very difficult to target to the right location in vivo. And her work has really enabled this, and it's been very exciting. So we're able to work together to develop therapeutics for vitiligo. This is Chi Tang, who is a shared postdoc between our labs, who's been working on this. This is some really intense chemistry of RNAi. She's modified the RNA backbone to make this completely resistant to RNases. And then also been able to link what we call a dianophore, or a hydrophobic molecule, onto the RNAi to allow self-delivery into the cells, the target cells, as well as the tissues of interest. So this technology allowed Alnylam to develop inclisiran, which targets cholesterol, and they found that this is now FDA approved, and a single injection of this siRNA is able to lower cholesterol for 6 months in patients. So this is very long lasting. And they were able to deliver this specifically to the liver. So we looked at their chemical modifications that they were able to create. These are different organs, and these are different chemical modifications of the drug with different dianophores, showing some will take the drug into kidney and others more into liver. But we were able to find one that actually was able to deliver to the skin pretty effectively. And so we know that interferon gamma blockade is an effective therapy in our mouse model, and likely in patients. And there's a human antibody called Gamifant that targets interferon gamma signaling. It's used for HLH. But there are a lot of reported infections with this antibody. And I know you've heard about an approach earlier with planned clinical trials targeting interferon gamma. I'm really interested to see the results of that trial. But we were asking whether there was a possible way to target interferon gamma signaling and keep it more local. So this is DCA conjugation. This is a hydrophobic molecule conjugated to the siRNA. We compared this to other conjugates as well. We found DCA actually was the best to silence interferon gamma receptor 1 in the skin compared to other chemicals. And so we proceeded with that. We can show we conjugated the siRNA to red fluorescent molecule. So you can see this is just injection into the tail with much better retention with the DCA siRNA compared to the unconjugated siRNA. And this is visible down here, where you can see the siRNA actually getting delivered into all of the cell types of the skin, including keratinocytes, fibroblasts, and many others. We were able to use flow cytometry to look at that delivery. We were getting close to 100% of the cells receiving the siRNA in both the epidermis and the dermis, and this included immune cells, keratinocytes, melanocytes, fibroblasts, endothelial cells, all receiving pretty good transfection with the siRNA. So Chi was able to show that we could get interferon gamma receptor silencing here. This is RNA, showing that it lasts at least 3 weeks, and then the expression started to recover by 4 weeks. When we looked at protein, we could see nice silencing of the protein for over a month, actually, with just a single injection. Here then we looked functionally, actually 2 doses of the siRNA against the interferon gamma receptor in the mouse tail. Then we took the tail and tail skin and stimulated it with interferon gamma, and were able to show that the siRNA delivered in vivo was able to turn off CXCL9 and CXCL10 expression, showing that the siRNA got into the skin and really silenced these pathways. So we're super excited about this, and we're testing it in vivo in our mouse model with plans to bring this into patients, hopefully, in the near future. We've also shown excellent delivery in human skin as well. So finally, a final story. This is really a project that was driven by my graduate student, Chow, who is an antibody engineer, and came to the lab and said he wanted to create a drug for vitiligo. And I said, well, I remembered an idea that John Stanley had at Penn when I was a resident with him, to develop a skin-tethered bispecific antibody to treat vitiligo. John studies pemphigus vulgaris, and he was able to pull out an antibody out of one of his patients called PX44 that binds desmoglein 3, but does not cause blisters. And so he came up with this idea that we could maybe take this desmoglein 3 binding antibody and fuse it to a therapeutic antibody and tether it to the skin so that it actually remained in the skin, unlike an untethered antibody that diffuses out. So, long story short, Chow was able to make these antibodies. This is the bispecific that binds desmoglein 3 as well as interferon gamma neutralizing. This is the desmoglein 3 binding alone and interferon gamma binding alone. And he showed that the bispecific antibody neutralized interferon gamma as well as the parent antibody. And it also bound to keratinocytes, both human and mouse keratinocytes, as well as the pemphigus antibody. So he created the antibody that does what it's supposed to, and then he labeled it with radioactive iodine and injected it into the foot pad of the mouse, and you can see that this unconjugated antibody, this parent antibody against interferon gamma, diffuses quickly out of the foot pad and becomes systemic in the bloodstream of the mouse, whereas the bispecific tethered antibody actually stays in the foot pad, comes out a little bit, and then goes back into the skin, and really remains at the site of injection long term. You can see that here, bispecific remaining in the foot pad, but not in the circulation. This is a movie that he took of the mice. You can see that the non-conjugated antibody, the non-tethered antibody is everywhere. You can see it's here in the circulation, in the heart, in the blood, whereas the tethered antibody is not. So this remains at the site of injection. Finally, Chow found that the injected foot pad here was protected from vitiligo compared to the other foot pad. And so he showed that the antibody remained where it was supposed to stay and did not affect the uninjected foot pad, whereas the untethered antibody actually protected the entire mouse, protected both foot pads equally. This is like a systemic injection, sub-Q injection of a biologic. And then the anti-desmoglein 3 antibody didn't protect either foot pads. So only the tethered antibody actually stayed in the one foot pad and protected that compared to the others. So we're excited about all the opportunities for targeting all of these pathways and potentially discovering new ones. We talked about JAK inhibitors targeting interferon gamma and IL-15 targeting for more durable therapies. We're optimistic that conventional antibodies, bispecific antibodies, next-gen siRNAs can be used for this to develop new treatments. So that's it. These are the people that do the work. I really have to give them excellent credit. I don't actually work at the bench anymore. They keep me out of the lab. It's a wonderful team to work with. We actually have a lot of fun together. My collaborators who have allowed all this, and we're excited to do even more. So that's it. I don't know if there's time for questions. I'd be happy to answer some if we have that.
Thank you very much, John. Professor Harris, for this fantastic lecture. You have opened a new windows for the future, listing all the trials or the different possibilities to think about the therapeutic approach of vitiligo. And this story that you had about the role of Treg is absolutely relevant for the pathogenesis of the disease, but just now listening about this possible opportunity for vitiligo, this opened certainly a new view for the future. Any question, comments, consideration from the audience? Please.
Hi, John. This is Amit. It was a wonderful lecture. I enjoyed it as usual. I have a question for you. During this conference, we've had actually quite a few talks about how to stimulate melanogenesis. Your lecture has really concentrated more on the immunology and trying to control the immunology. What ideas or developments or pathways have you opened up or had for the other part, which is really to try to stimulate melanogenesis? Yeah.
I don't intend to say that that's not important. It's something that we haven't focused on in our group. We've really, as an immunologist, have focused on the immunology. I think that if we can stimulate melanocytes and or protect them from cellular stress, as Dr. Picardo was presenting yesterday, that is a beautiful kind of pairing that would, in addition to the immunosuppression, would be a much better treatment. We know alpha-melanotide actually is effective through clinical trials. This is an alpha-MSH analog stimulating the melanocytes to regrow. When paired with narrowband UVB, actually accelerates repigmentation. I know that Clinuvel is launching a new clinical trial to test that again. Zalfa Abdel-Malik is working on developing peptides that will stimulate melanocytes potentially topically. We've talked about antioxidants. We haven't found kind of the right mix yet, I don't think, to protect melanocyte stress. But if we could do that and combine the whole thing together, I think we'd have a really phenomenal treatment. So I think with the JAK inhibitors, this is just the tip of the iceberg, and we've got a lot of work left to do.
Yeah, and during this conference, we had a presentation from AHA-Immune about a gel. It's a non-steroidal that's applied to guinea pigs that was able to stimulate tyrosinase and stimulate melanocyte activity with a gel that at least it looks good in animal studies. So there's some excitement going on.
And I saw Dr. Ramaya speaking as well earlier. He's been working on decapeptide. There are lots of ways to really stimulate melanocytes. We have to find something that's going to work in vivo and create that really important tool in our toolbox. And thanks for all your help, Dr. Pandya. We've got your name's on a lot of our papers and has been really essential to help us learn about pathogenesis.
Dr. Harris, I'm Charlene, Dr. Ng from Taiwan. Thank you for a wonderful talk. Yeah. Yeah, so my questions, there are two questions. First one is about the anti-IL-15 and the anti-IL-15 receptor antibody. Which one would you think it's better work and in terms of the safety and durability and stuff? The second thing is about the anti-interferon gamma antibody. So the study is done in HLH, which is a group of patients which is more immunocompromised. So it seems like the whole story actually circulates around interferon gamma in the beginning of the initiations, and just study from the, I think, Beijing supports that the fibroblasts also, and your studies increased in interferon gamma. It's a very critical pathway. So my second question is, do you think the anti-interferon gamma antibody might work in vitiligo because they're not like as immunocompromised in HLH, and studies in SLE and Crohn's disease seems to not like very elevated. Yeah, thank you.
Yeah, I saw your talk. I was really intrigued by that. I'm excited to see what comes out of that study. I've been wanting to block interferon gamma to treat vitiligo for about 10 years or more. And actually looked to partner with pharmaceutical companies, and the drugs just weren't available. So it sounds like you've got an opportunity that we didn't have. And you know, HLH being more immunocompromised, they may be more susceptible to infection, and we see this in other drugs as well. When you treat healthy populations with biologics, we don't see the same side effects. So I think it's a great idea, and I think it'll work. So I'm going to keep a close eye on that. The targeting IL-15 cytokine versus the receptor, one of the challenges is the IL-15 cytokine is expressed in every tissue of the body, of the human body. So the protein burden is really hugely significant. So it's very hard to develop an antibody that will saturate that signal and turn it off. That's why we think targeting the cytokine probably isn't going to be very effective. We know that Amgen's antibody, of course I'm biased, right, because I started a company that's competing with it, but Amgen's antibody has been in clinical trials in humans, and it doesn't affect NK cell number. NK cells actually rely on IL-15 as well. So when you successfully block IL-15, NK cell numbers drop in vivo. We've seen that with other studies. And we see that in non-human primates when we use our antibody to target the receptor alone, which is only expressed on T cells and NK cells. So a much easier approach to try to saturate that signal. But we'll see. You know, the trial's ongoing, the REVEAL trial's ongoing. Our trial with Insight will hopefully be started soon. So hopefully we'll have data.
I have a question, Professor Harris. Just speaking about the possibility of the repigmentation, probably we don't know so much if the inflammatory milieu that is certainly present in the skin of vitiligo patients, probably also in pigmented skin, can control the capability of precursor of the melanocytes to start with the possible differentiation process. Do you have any idea or some eventually experimental evidence that changing the situation with this new type of treatment, that is probably something that we have started to see with using JAK inhibitors, but probably adding different type of therapies, as you suggested, the combination to inhibit different steps of the immune response spontaneously can restart the possibility to regenerate. No, probably we have to regenerate melanocytes during our life. Normally, it should be some, change. So we will have some mechanism which precursor melanocytes start to replace the old melanocyte. Do you believe that per se, changing the situation, blocking out interferon gamma mediated mechanism of the chemokines or interleukin-15, the pigmentation can start spontaneously, or do you believe that we need something to stimulate this pigmentation?
Yeah, it's an important question. I think so. My bias is that I think melanocytes are constantly trying to repigment the skin. The T cells are constantly killing them. And so if you can shut off the inflammation, I think the melanocytes already know what to do. They're capable, as you said, they know how to heal wounds, they know how to repigment wounded skin. I think they're ready to repigment, and it's the immune system preventing that. We don't know for sure, and we know that narrowband UVB helps JAK inhibitors work even better. And so there's probably some level of repigmentation that's spontaneous, but it certainly can be helped out a lot. But we'll have to see. The INSIGHT clinical trial was just a JAK inhibitor with no phototherapy and no intentional sun exposure, and that alone worked. So I kind of think it's spontaneous, and those melanocytes know what to do, but we certainly can help them out, I think.
Thank you. Any other question or comments?
Mauro?
Otherwise, we will...
Yeah. Mauro?
Yeah, sorry.
Hi, John, it's Thierry. Very great job, as usual. I have one comment and one question. The comment is on the melanogenesis. In fact, it's very important to differentiate melanogenesis and proliferation of the melanocyte and the differentiation of melanocyte. Because if there is no melanocyte, you can use whatever you want for stimulate the melanogenesis, it won't work. So, I mean, especially in some areas, and we see that with the JAK inhibitors trial, that there are some areas that are very difficult to differentiate. And I do think that in those areas, we need to stimulate the differentiation of melanocyte stem cells. For the question is that on the Tregs, I enjoyed this story. You show that, if I well understood, that you had a defect on the Tregs. You have more Tregs coming into the lesional areas. Usually Tregs are against and are decreasing the CD8 activities. So what is your opinion? What is your thought about that? Do you think that the Tregs are there, but they are not enough, or they are functionally altered?
Yeah, I didn't have. I'm glad you asked that question. I didn't speak too much about that. I think the first thing that you said, the former, that there are not enough Tregs. So the Tregs are trying really hard, and in that paper, what we found with the non-lesional skin, the Tregs were in there and they were turned on. We could see that they were activated, they were responding to interferon gamma, they were working really hard in non-lesional skin, and they were being successful. But in the lesional skin, actually, then there's way more CD8 T cells. I think they overwhelm the number of Tregs. More Tregs come in to try to control them. And actually, the Tregs are turned on in lesional skin, too. So we see them just as turned on just as much in lesional skin as non-lesional skin. So it's not like they give up or they turn off, and that allows the lesions to happen. It's that they get overwhelmed. And they're just not, their function is active, and they're just not, it's not sufficient to control all the CD8s that are there. I know this is a bit controversial. There have been a lot of studies in Tregs in vitiligo. Some studies have shown that there aren't enough Tregs in the skin, that they don't get into the skin, or that they're non-functional in the skin, or others have shown that they're non-functional in the blood. But really what we saw is they're working, they're trying, they're working in the skin, they get to the skin, they're just overwhelmed.
Yeah, we have another question.
I agree with the melanocyte stuff. So there's a lot to melanocytes, and again, it's not our expertise in the lab. So I think that we need really, to think widely about how to get them to differentiate and proliferate and migrate and make pigment. It's all important.
Thank you for your great lecture. I'm Dr. Choi from South Korea. In many researches, saying that vitiligo is a systemic disease, so I think the increased interferon gamma or CXCS10 or 9 in patient blood can activate or sensitize the immune cells in the system and then makes a new region in another site. So do you think that the local treatment using RNAi or by tethered antibody can control or halt the disease progression in another site?
No, probably not. I think that the local treatment, we know that too. We know that with like narrowband UVB, you can control an area, an exposed area, but it doesn't affect another area. So if they're wearing, you know, a t-shirt, any place that's not exposed to the narrowband UVB will continue to progress. And this tells us really that local therapy stays local. I don't know how much, you know, vitiligo is a systemic disease. We definitely see Hashimoto's thyroiditis and other autoimmune diseases occur more often. And I think that's because of shared genetic risk. If you have vitiligo, you've got a bunch of genes that cause vitiligo, and those same genes also cause thyroid disease. Of course, you have Vogt-Koyanagi-Harada syndrome, where patients with vitiligo then get inflammation of the brain, the meninges, the eye, the ear. Melanocytes are in all those sites. I don't know that I would call that systemic disease as like multi-organ targeted disease, because those melanocytes are there as well. But it's hard to know. I think there's still a lot to learn. So if we do find that it's systemic and that the interferon gamma in one area of skin is driving disease elsewhere, I don't think that the local therapies will help the other locations.
Other comment or question? Please, another one. Ilfat.
Thank you, John, for your excellent lecture. We're really sad you're not here, but hopefully in Cairo. So we're giving you advance notice, December 2024.
It's already on my calendar. Patrick told me about it.
I'm picking you up. So, John, there's also some conversations over the meeting, some really nice posters about the role of fibroblasts in vitiligo. So can you comment on fibroblasts in that particular cell line?
Yeah, so Mauro's been talking about fibroblasts for a long time, and I've learned a lot from him, you know, in their role and how they support vitiligo. And then there's the paper that was published just this year in Nature showing that fibroblasts make the CXCL9 and CXCL10 as well. I guess that's not too surprising. They respond to interferon gamma. So fibroblasts, I know Mauro's work talks about fibroblasts and stress and you know, the kind of the non-immune function of fibroblasts. I think they function in an immune way as well. So they probably are in a position really to influence disease, both in recruiting cells into the skin in the first place, and then also kind of responding to the melanocyte disruption as well. So there's still so much to know. You know, there's a lot of cells in the skin, and we've only, I think, scratched the surface in how they all kind of work together to drive disease. The single cell sequencing really taught us a lot. We were surprised. We did that just to try to see what T cells were doing. But we were able to pull out keratinocytes and Langerhans cells and macrophages, and they're all doing something. That ligand receptor map that I showed, everybody's turning something on and contributing some way. So it's really a very complex conversation that drives that, I think.
Any other comment or question? This is just an open discussion with Professor Harris. No? So we thank you a lot, Professor Harris, for your talk, for this discussion.
Thank you.
We missed you yesterday with your comments during a lot of very interesting communication that we were capable to follow about immune responses, but certainly we learned a lot with your talk. Thank you very much.
Thank you.